My name is Laura Johnston. I'm the Scientific Associate Director at the Flow Corps at UChicago. This video is the first in a series that will go over the practical steps of how to perform an actual flow cytometry experiment. These videos are intended for users who already have some knowledge of flow cytometry. We do have a Flow Basics course that is required to use our facility at UChicago. So really, this is a follow-up to that original course. My goal here is to break down the practical steps of running a flow cytometry experiment and help you understand how each of these steps could ultimately affect your final results. So in this first video, I'm going to go over the basic staining protocol. And then the second video, I'm going to build on this first video and discuss how to optimize the protocol. The third video will go over panel design. Fourth video, we'll talk about experimental design, which is mainly choosing controls. And then the fifth video, I'm going to talk about some things you might want to think about while you're actually sitting at the instrument, like how to set up the instrument and how to utilize automated compensation. So as I mentioned, I'm going to talk about a basic protocol for this video, mainly intended for those of you who have little to no experience staining. I'm just going to talk about surface markers here. And again, the overall theme of this is how to improve consistency in your sample staining. So let's get to it. The first thing before we actually get to the protocol is I want to talk about staining tubes and buffers. Really, the choice is up to you. You can stain in whichever tube or format that you want. The main difference has to do with wash volume. So common tubes used are Eppendorf tubes, 5 ml round bottom tubes, which are often called fax tubes, or a 96 well plate. Usually the round bottom plates work well. And again, you just will wash your cells in a different volume depending on which tube you're using. So in an Eppendorf tube, you can wash with 1 ml. In a fax tube, you can wash anywhere between 1 to 3 mils, and then obviously a 96 well plate can't hold that much, so I usually do around 200 microliters per well. Another thing to keep in mind with your staining tube is that some instruments require a specific tube type to run your sample. So it's up to you if you are okay with transferring your samples from one tube type to another. So if you wanted to stain in Eppendorf tubes, then use a Fortessa. You would have to transfer your sample from the Eppendorf tube to the fax tube. Or if you don't want to bother with transferring, then you're going to have to know which type of tube is required for the instrument that you're planning on using. I do have the catalog number here for the 5 ml fax tubes that we use on our Fortessas. So if you are using the Fortessa, then make sure you order this specific tube type because it does need to pressurize properly. Then for buffer, it really depends on your cell type, whatever keeps your cells happy. But there's kind of a lot of options. Typically for fax buffer, our base is 1x PBS. We usually do calcium and magnesium free. The reason for this is that it's very similar to the sheath fluid in the instruments. But if you wanted to use a different type of buffer, I don't entirely see a reason why not. There might be a few minor issues, but you should be okay. The main issue would be if you had phenol red in your buffer. So just make sure that it's a clear buffer. The phenol red can get excited by the lasers in the instrument, so you don't want that. In addition to the PBS, we recommend adding some protein to make sure your cells stay happy. So usually FBS or BSA. I have the recommended percentages for those listed there. Again, there's a range. It depends on your cell type. The one that I typically tend to go with is 1% FBS. And a note about if you're using FBS, especially if you're doing cell sorting, FBS does have some debris floating around in it. So if you are doing sorting specifically or really anytime. It's a good idea to filter your FBS beforehand so you remove that debris from the FBS because you'll pick it up on the cytometers. Not the end of the world if you don't do that, but it certainly helps things. Then beyond that, there are other additives that you could use. Not everyone uses them. Some people will add EDTA if you have really sticky cells. Some of the vendors will add sodium azide to their buffer as well. You don't have to. You're fine with PBS and FBS or BSA, but just know that sometimes there are other additives. Okay, so once you've decided on your tubes and your fax buffer, which is used for washing and staining and pretty much everything, then you can start doing the protocol. So this is just an overview of the basic protocol. The first thing you're going to need to do is acquire your cells as single cells in suspension. So if you have tissue, you're going to need to get those out of the tissue. I'll talk about that a bit more in the second video. And then you will add Fc block to help with nonspecific or unwanted staining. Then your fluorescent antibodies, and then once those antibodies are bound, then you will wash away any of the free antibody and run it on the cytometer. So the first thing I'm going to talk about is the Fc block. There are a variety of ways that antibodies can bind to cells, but for our experiments, we only want the antibody to bind in one way. So here is an example of that. We've got our antibody here. I have a little red dot that is for the fluorophore, and you can see that the variable regions of the antibody are binding the marker of interest. So this is the way that we want our antibodies to bind to our cells. However, most of the antibodies that are used for flow cytometry are IgG isotype, and a lot of immune cells have receptors for IgG antibodies against the constant region. So this is still a specific method of staining. We have an IgG receptor that specifically binds to the Fc portion of the antibody, but for our purposes, it's not specific for our marker of interest, and therefore we don't want this type of staining to occur. So we add Fc block to prevent this type of staining. It's something that you can just buy from manufacturers. It's not too expensive, and it will block this binding quite well. And then another method would be non-specific staining, which I've shown over here. Typically, I find that just having some protein in your staining buffer is enough, but if you want to go beyond that, you can certainly add additional serum if you would like. In the microscopy field, they tend to add rabbit serum for rabbit antibodies, goat serum for goat antibodies, etc. If you wanted to do that, you're welcome to do that. It's up to you. Or you can also increase the percentage of protein in your staining buffer. That may also help. In my experience, I haven't done any additional serum, but just keep in mind it's something that you could do. So once you've added your Fc block, incubate that for about 10 minutes in the fridge, and then you can move directly on to your antibodies. And again, I would check the manufacturer protocol for what they recommend, but this protocol tends to work well. So now I'm going to talk about how you would stain for surface markers using your fluorescent antibodies. There are certainly other markers beyond this which would require more steps. For example, if you're staining intracellular markers, then you would need to permeabilize your cells first. But for this talk, I'm just going to talk about surface staining. There are 5 factors that will affect how your antibody stains, or essentially your results. Those factors are time, temperature, volume, cell number, and antibody amount. Now, as I mentioned in the beginning, we want to keep our experiments very consistent so that we can compare our experiments and interpret them appropriately. If you alter one of these factors between experiments, then you may find that your results are different due to the staining protocol that you altered and not due to any biologic results. So it's really important to keep in mind all of these factors that can alter your staining and keep them as consistent as possible. For time and temperature, these are pretty easy to keep consistent, so I would say just pick a time and temperature and always use that. So I tend to do 30 minutes at 4 degrees Celsius. I've seen between 15 minutes up to an hour. In rare cases, some antibodies might need to be incubated overnight. Sometimes people will stain at room temp instead of 4 degrees. So again, I do recommend reading the manufacturer protocols, but typically you can just pick a time and temperature, stick with that, do that for all of your experiments, and in rare cases, for some antibodies, you might need to alter that. But for surface markers, in my experience, I always do the same thing and it generally always works. For staining volume, the default here for us is 100 microliters. You can obviously change that, but for me, I always stain in 100 microliters. I don't really stray from that much. In the second video, I will talk about how you might go about changing that if you need to, but our starting point is generally 100 microliters. And then finally, cell number and antibody amount. Obviously, these depend on volume a bit because really the important thing is cell concentration and antibody concentration. But to start with, 1,000,000 cells is a good place to begin with. And for an antibody amount, generally 0.1 micrograms of antibody, which depending on the concentration of your antibody, oftentimes works out to be somewhere between half a microliter to a microliter of antibody. But we will be talking about this in more detail in the second video because using this default starting point is often not what will be best for your experiment. But if you have no idea where to start, I would say start here. So in addition to these 5 factors, I also want to talk about pipetting because that's another source of variability that you could get in your staining protocol. So I would say avoid pipetting small volumes of antibody. Usually that means less than 1 to 2 microliters. So if you need to pipette less than that, I would recommend making a 1 to 10 dilution of your antibody first. So I generally dilute 2 microliters of antibody into 18 microliters of Fax buffer. And then you can pipette 10 times as much, and it's a much more reasonable volume to keep consistent between your samples. Now, if you have multiple samples, multiple antibodies, then we do recommend that you make a master mix of your antibody cocktail, and that will ensure that your samples are stained with the same amount, and it will minimize the amount of pipetting that you have to do as well. So I'm going to go through a little example of how I might go about making a master mix and my actual protocol for mixing everything together. Over here, you can see in this table, I have the amount of antibody that I would use per sample. So let's say I've done an experiment, I figured out how much antibody I need. So for each sample, I would need 1 microliter of my FTSE antibody, 5 microliters of the APC antibody, and half a microliter of the PE antibody. So, if I had 8 samples, I would have to pipette half a microliter into 8 tubes. That would not be very consistent, most likely. So instead, I like to make a cocktail and then bring that up to a total of 50 microliters. So I'll add 43.5 microliters of Fax buffer to these antibodies. So then I would just need to add 50 microliters to my actual sample. But let's say I have 8 samples to run. So then I'm going to actually instead of pipetting individual amounts, I'm going to make a cocktail. So with 8 samples, I'll make enough for 10 just to account for some error in pipetting. So I would add these volumes together to get a total of 500 microliters, and now I can take 50 microliters from this master mix cocktail of antibodies. So to actually do the pipetting, what I like to do is resuspend my million cells in 50 microliters of Fax buffer, add my Fc block to that 50 microliters, incubate that for 10 minutes, and then add 50 microliters of master mix to my 50 microliters of cells, and that brings up my total staining volume to 100 microliters. So I essentially do my Fc block in half the volume, and then my master mix is essentially a 2x concentration. So that's my preferred method. You don't have to do it this way, but I find this to be the most straightforward. Another note, once you add your fluorophores, you want to make sure that you protect those from light because they will start to photobleach once they're exposed to light. That generally means that we'll just cover the rack of tubes with foil and then incubate them somewhere dark. So depending on whether or not your fridge has glass in it or not, then you need to be more or less careful with the amount of foil that you're covering your tubes with. If I have my tubes sitting at room temperature, I generally put them in a drawer or a cabinet just so it's a little bit darker. And then another note to keep in mind about this is if your cells themselves have fluorescent proteins, then that means that they should always be protected with some foil. Otherwise, if you don't have fluorescent proteins, then you only have to worry about this once you add your antibodies to your samples. So once you have incubated your antibodies for whatever time you have decided on, then you just need to add some Fax buffer to your tubes. Again, depending on which tubes you're using will depend on how much volume you add to each sample. Centrifuge those and then aspirate the supernatant. It's up to you how you want to get rid of that supernatant. If I'm using a 5 ml round bottom tube, I tend to dump it. So if you just in one quick motion dump the tube over, that tends to work quite well and is very fast. If you do have a vacuum to aspirate the supernatant, just be careful about sucking up the pellet. And then now you are ready to resuspend. So that's another question I get a lot is what volume do I resuspend my cells in? It really depends on the cytometer and also how comfortable you feel running it. So if you're newer, then it is safer to resuspend your cells in a slightly larger volume so that you have enough time to set up the instrument before you run out of sample. And as you become more comfortable, then you can certainly decrease the volume just so things run a bit faster. So for the Fortessas, we recommend that 150 microliters is the absolute minimum volume that you would ever want to bring. Again, if you're newer, we would recommend bringing no less than 300 to 500 microliters in each of your tubes. Again, if you feel more comfortable with the instrument, you can certainly decrease that down. But in general, you would not want to run your samples faster than 35,000 events per second. Sorry, that's a typo. Shouldn't be less than, should be more than. For the attune, we have a chart on our website, so make sure you check out that chart. And then for the sorters, the absolute minimum is 500 microliters. And then the concentration, the maximum concentration recommended is dependent on the tip. So for the 70 micron tip for smaller cells, then no more than 20 to 25 million cells per mil. The 100 micron tip is no more than 10 to 15 million cells per mil. And last, I just have a couple additional notes. I want to talk about viability stains. Sometimes I get questions if people are familiar with microscopy where they tend to use primary and secondary antibodies. I'll touch on that subject, and then also fixatives. So viability dyes are very important. You definitely want to include those in your experiment to remove the dead cells. But the protocol depends on which type of viability dye you choose. So if you choose a DNA binding dye, which is DAPI or PI are the common ones, they're more beyond those. But those dyes you would want to stain after you've completed the protocol. So after you stain your antibodies, resuspend your cells, you're ready to go, you're about to leave your lab and walk over to the flow core, you would add your DAPI or PI to the tube, and then you're ready to run in the instrument. You don't need to bother washing or anything like that. The downside of these dyes is that they are not fixable. So if you need to fix your cells, then you can't use DAPI or PI. Instead, there are some fixable dyes. So BioLegends are called zombie dyes, or you can get the fixable live dead dyes from Thermo Fisher. They're pretty interchangeable. The way that these work is they bind proteins through amine groups. So on live cells, they will bind surface markers or surface proteins, and then on dead cells, they will bind intracellular and surface proteins. So the dead cells will be much brighter. The difference is instead of staining at the end, like you would with the DNA dyes, these need to be stained at the very, very beginning. So before antibodies, before FC block, you would stain with your viability dye, and then it also needs to be in a separate step because it needs to be in a buffer that is free of protein. So no BSA or FBS, just PBS only. So it doesn't matter which viability you use, just keep in mind that there's differences in the protocols. And then for the subject of using a fluorophore conjugated primary antibody versus an unconjugated primary antibody plus a fluorophore conjugated secondary antibody, we tend to, as flow cytometrists, keep things simple, so there isn't really a benefit of using a secondary antibody. If you use a primary antibody, it's less incubation time, less optimization, fewer chances for nonspecific or background staining, so we recommend just using the primary antibody. If you want to use the secondary antibody, you can if you want to. Just keep in mind that you need to titrate both antibodies, and it's going to be extra steps and extra washing, extra chances for background staining. But you can certainly do it if you really want to. And finally, for fixatives, if you have BSL-1 cells, you can run those fresh on all of our instruments. If your cells have infectious particles in them, so they're BSL-2 or above, then those do need to be fixed before you run them on our benchtop analyzers. Now, there are a lot of different options for fixatives. It's entirely up to you what you want to use to fix your cells. But I just want to point out a couple of concerns that might occur with fixation that you should be aware of. So if you fix your cells at the very beginning and then stain them with antibodies, you run the risk of the fixative altering the epitope of the marker that you're trying to look at. So that means that your antibody might not stain at all, or it might stain differently than it does when you're staining fresh cells. So just keep that in mind. If you're fixing at the very end, so you run through the entire staining protocol and then you fix your cells, just know that there's a chance that some of your signal intensity might decrease. So if you're comparing experiments, you definitely want to keep things consistent. So if you're fixing cells, fix for the same amount of time, the same type of fixative. Don't compare unfixed cells versus fixed cells because you're probably just going to see differences in intensity due to the fixative. And then finally, the fixative could potentially increase the cell autofluorescence, which would mean that it's a little bit harder to identify your positive population on some of your markers. So, these aren't necessarily huge problems that are always going to make your data terrible, but just something to keep in mind that you might find that this happens in your experiment, and certainly you want to keep the fixative the same if you're trying to compare experiments. And I just have a couple links here regarding fixation that you might want to check out if that's something that you're interested in doing. Again, you can download these slides on our website. That's it. Stay tuned for the next video in the series where I will build on this protocol and talk about how you would actually go about optimizing it.